All right, you all. Good morning. It's Saturday, but I'm using my meowgical mug. It's my daughter's. She'd be mad at me if she knew I was using it. But I hope you guys are having a good weekend so far. One day is the same as the next, right? So I just want to finish up male reproductive behavior because this has been going on for way too long. So this is going to be quick, just talking a little bit more about the neurochemistry that we know of male sexual behavior. One thing I want to point out is, as I mentioned in class, almost all of the research has been done in rodents. So what I'm going to talk about is some of the connections that we know of because of rodents. So olfaction is a major player for rodents. I have this picture here of the rat brain because you could see this protrusion right here sticking out of the rat brain. That is their olfactory bulbs. So if you think back in 295 when you did your brain dissections and you remember looking at the olfactory bulbs, hopefully you guys actually saw the size of the olfactory bulbs in the human, very tiny compared to the brain. But you can see that for rats, the olfactory bulbs are huge. This is how they navigate their world. So olfaction to them is like vision is to us. Large parts of their brain dedicated to this sense. So what we know about rats is that they have something called the vomeronasal organ, or the VNO. This is something that is debated about whether it is something that is, whether it's something that we have, if it's a part of the brain that we have. Some studies suggest that we do have one. It's just really tiny. Some studies suggest that we don't. So it's a little bit unclear at the moment if we actually have one of these organs. I do believe that we probably do have one, but again, if we do, it's really small and it's unclear what the role is for us as far as pheromones and stuff like that. But for rats, pheromones are incredibly important. So the vomeronasal organ is really important for male reproductive behavior in rats. This has a lot of connections to the medial preoptic area, which, as we talked about, is the nucleus that is responsible for male sexual behavior in the hypothalamus. The other thing I want to point out with the vomeronasal organ is that rats can't tell the difference between male and female rats unless they're able to smell them. So this organ is important for sex identification in rats. And you can see here, this diagram here shows the olfactory bulbs, and you can see where the medial nucleus is in the hypothalamus. Over here is the medial preoptic area in the hypothalamus, sorry. And then we're going to talk a little bit about the amygdala as well, because that is playing a major role in male sexual behavior in rats. So here is the olfactory bulbs here, and then the vomeronasal organ. Okay, so in order for, unlike humans, rats actually females have to be in estrus in order for mating to take place. So it's really important for a male to be able to identify when a female is in estrus. We'll talk about estrus a lot when we get into female sexual behavior. So essentially what we know is that when a female is in estrus, that scent will be received by the olfactory bulbs, and this will cause the neural response in the medial preoptic area. When this happens, then gonadotropin releasing hormone will start to be released, and testosterone will be increased. So this causes sexual behavior in the male rat. So if you have a castrated rat, then those electrical signals are no longer occurring. So there is something important with testosterone and the olfactory bulbs, and the circuit needs to be in place for this to occur. Okay, so a bulbectomy is when you remove the olfactory bulbs from so here's the vomeronasal organ here, and then here is the brain, here is the olfactory bulb up here. You can see here, this area is the olfactory bulb in the rat brain. So when we take out the bulbs, and we perform this bulbectomy, then we do see both the appetitive and the consummatory behaviors be reduced. It depends on how much sexual experience the rat has had. Sometimes the rat will still engage in sexual behaviors, but they are certainly reduced in some of those rats. Again, this isn't an end-all be-all about it. But it seems that what is the important part of all this is the nerve from the vomeronasal organ. So the vomeronasal organ will receive the pheromones and will send those signals into the brain, into the main olfactory bulb. So if you destroy this nerve, you get the same effect. So it's not the bulbs themselves. It seems to be the input that it is receiving from the vomeronasal organ that is responsible for these behaviors. Okay, so let's talk about the amygdala for just a moment. And again, what I'm talking about is for rats in particular, but in general, the amygdala does tend to be larger in males. We didn't really get to talk about brain sex differences between males and females. Hopefully we'll get around to that at the end of the section. But the amygdala is also receiving input from these olfactory bulbs and from the vomeronasal organ. So the amygdala has has 12 different nuclei actually. And so it does seem that certain nuclei in the amygdala are responsible for certain behaviors. The amygdala is mostly important for fear and aggression. So it's attributed to emotions, but in particular, it's fear and aggression. If you experience more fear or less fear, you're going to have different behaviors, or more aggression, less aggression. So it's actually fear and aggression that the amygdala is really important for. If any of you guys have taken 300, I assume you guys have talked about that. So this particular nucleus, the basolateral nuclei, and I don't have a picture of the amygdala on here, sorry about that, but if you remove this individual nuclei of the amygdala, then you're going to decrease appetitive behaviors, but consummatory behaviors are still going to be intact. If you take out the corticomedial nucleus, then you have ejaculation latencies in the rat. The amygdala does send information back to the preoptic area as well. Okay, so let's take a moment and talk about receptors in the brain. So many studies have suggested that the receptors, the location and the density of androgen receptors and estrogen receptors seems to be similar in rats and primates and probably humans. So this seems to be pretty well conserved in mammals at least. However, it's important to note that these behaviors are a lot more stereotyped in rats, where humans can engage in sex in any time. And even if a male has had a vasectomy or no longer wants children or is using a condom or something like that, he obviously can still engage in sex. So these behaviors are still taking place even when reproduction is not the goal. So human sexual behaviors are a lot more environmental. There's a lot more external stimuli that seems to be playing a role. So it's a lot less associated with hormones and sex receptors compared to rats and primates, non-human primates. But nevertheless, the density and location of receptors is pretty well conserved between different mammals. So there are different receptors in the brain. We have androgen receptors, which are abbreviated as ARs. We have ERs, which are estrogen receptors, and they are going to be different categories of receptors. We have ER alpha and ER beta. These are the two types of estrogen receptors found in the brain. And then we have progesterone receptors, PRs, and there is high densities of all of these receptors in the hypothalamus, as you can imagine, since the hypothalamus is so important for reproductive behaviors. So we talked about the three different types of estrogens. E2, estradiol, is the one that is the most prominent. It's the one that we know the most about. It's the one that is high during our reproductive years. So this is the one that has received the most research, and it does seem that estradiol is going to bind to ER alpha and ER beta to the same degree. Estriol E3, this is the one, oh my gosh, I hope I don't mix these up. This is the one that is occurring, I think this is the one that occurs in pregnancy. I might get these mixed up. One of them occurs in pregnancy and one of them is higher after menopause. So estrone E1, I believe, is the one that is happening at menopause. And that one seems to have a higher affinity for ER alpha, and estriol E3 seems to have a higher affinity for ER beta. Okay, so as I just said, there's high densities of these all throughout the hypothalamus, particularly in the medial preoptic area in males, but you could also find these in other areas of the brain. You could find them in the forebrain. You could find them in the ventral tegmental area, in the reward pathways. You can find these in the midbrains. You can find these all over the place, especially in the limbic structures. You're going to find them in the amygdala. You're going to find them all over. Aromatase activity, though, this is important because this is going to convert. Remember, this is going to convert testosterone into estradiol, and you will see high activity, as you can expect, in the medial preoptic area, in the ventromedial nucleus of the hypothalamus as well, and you'll also see high activity of this in the amygdala. There's also activity, you will find these also in the hippocampus as well. When you castrate a male rat, you do see a decrease in aromatase activity. So this suggests that, that you're not going to get as much conversion of testosterone into estrogen. So what's interesting about this is that when you castrate, you will see this reduction in activity in everywhere, but you don't see it in the amygdala. So I don't exactly know what that means, but that's pretty interesting. Okay, so here's a little bit, we're going to talk more as we move on throughout the semester about the specific estrogen receptors and some roles that they play. So we can do knockout studies to see where these male sexual behaviors are coming from. Is it through ER alpha? Is it through ER beta? Or do you need both of them? Well, so it's really unclear in as far as male sexual behavior goes as to which estrogen receptor is the most important. You do see mild deficits, but it's mostly performance-based. For ER alpha, you don't see any changes in a knockout study with ER beta, but if you do take out both of them, then you eliminate all sexual behaviors. So you do need both of these receptors functioning to have full normal sexual behavior in males. But again, if you knock out ER beta, you're not going to see any deficits at all. Everything remains the same. And if you knock out ER alpha, you're only going to see mild deficits at best. But so it's unclear exactly what role each of these receptors plays in male sexual behavior. And that's all. So that was a really brief video. We are going to move on from male sexual behavior now, and we are going to start talking about female reproductive behavior. I hope to get another video up hopefully this weekend so we can start rolling on that. I did share some information in the chat about how I'm thinking about the course moving forward. I'm probably going to have our exam on the week of April 20th. I'm currently thinking maybe April 24th for that exam. We'll have to see how far we get. But if you guys have questions, I'll see you on Monday in Slack. See y'all.